Next speaker is from the Department of Zoology. Professor Derek Smith is going to pick up the theme of pathogens that we ended on in Alan Short's lecture, and is going to be talking about modelling emerging pathogens.
Yes, thank you very much. A little over a year ago, a group of people from many different parts of the university came together and were asked to come together by the WHO and were awarded the status of being a WHO collaborating center on emerging infectious diseases, control of emerging infectious diseases. What I'm going to talk about today is just a couple of minutes on a little bit of the structure of this center, and then 4 little anecdotes about things that people within the center have done, and a little bit about our plans for the future. This is still a beginning initiative. In many ways, it doesn't change the immediate work that many of the people who came together that were designated as the center do. But what it does do is it brings things together and helps to network people in the same way as was talked about at the beginning of here for the Public Health Network. And we are still in the process of, this is still a nascent activity that is coming together. In fact, on Thursday we have a meeting, a little bit analogous to this one, where people are coming together to discuss strategy for the meeting, for the center. To give you a little idea for the breadth, a couple of us from zoology, Sharon Peacock from the medical school. Whoops, I was thinking this was 2 pages here. It's 2 pages in the brochure. Sharon Peacock from the medical school, David Dunn from pathology, James Wood and Duncan Maskell from the vet school. So you see people coming from many different areas working on communicable diseases with a particular interest in diseases that cross species barriers, and also diseases that change once they are in humans, so-called emerging and re-emerging infectious diseases. As well as that, there are others involved from around the university and from these centers, Efrosini in the medical school, Jim Kaufman from pathology, Koen Stamers from architecture, Simon Frost and John Heaney from the vet school, Alan Blackwell from computer science. A very broad range of people, as I say. Let me give you sort of some examples and a little bit of an idea for sort of what a collaborating center might give one. One of the things that it gives one is a badge, and a badge to do some things in a way where perhaps WHO will come and say, can you help us with this? Where we might come up with either small technologies that we think might affect global health with strategies that are useful to do at a global level, and also sometimes access into WHO to come in with the sort of disruptive technologies that research can bring sometimes to global health. The first example that I'm going to give you is a disruptive technology that was developed in many places, well, not in many places, but predominantly here in Cambridge and published in 2004. This is a technique to look at variation among pathogens, not genetic variation, which of course is very important, but this is the sort of variation in the particular type of phenotype, the antigenic aspect of pathogens, the aspect that our immune system sees. And when pathogens change their antigenic phenotype, they escape. They become resistant to antibiotics, or they escape vaccines, or they escape immunity that we might have in the population. Measurements of these sorts of phenotypic differences have been very difficult to interpret at fine-grained levels. And this technology, called antigenic cartography, allowed a much finer and higher grain resolution interpretation of those data, such that one can much more detail, from a research standpoint, study the evolution of pathogens, and from a public health standpoint, apply this sort of work to multiple aspects. Today I'm going to talk about public health aspects. This is an example of an antigenic map from that disruptive technology. Each of the colored dots here represents a different strain of a pathogen, in this case influenza virus. And when dots are close to each other on this map, they are more similar antigenically. To give you an example of how what that would actually mean functionally, if somebody were vaccinated with a strain of flu that's in the middle of one of these clusters, one would expect them to be protected against all the other strains in that cluster, but not against strains in the next cluster. And so when the virus evolves from one cluster to the next through time, these numbers here are the year when that cluster first appeared, that's when the strain of flu that is in the vaccine needs to be updated. This is done sometimes on a yearly basis. fact, one of the questions of influenza virus strain selection, and also a very deep fundamental question in evolutionary biology, an evolutionary question is, if the virus, let's say, was in this particular cluster, where would it actually go next? You don't have to look for very long at an antigenic map like this to say, it'd be a pretty bad guess to say it's going to go here next or over here. It's likely to be in one of these directions, but which direction? How far is it going to jump? And also critically, when is it going to jump? Because that's what we would like to predict to have the right strain of flu in the vaccine. Soon after this work was published is when this particular work started with the World Health Organization. It was in 2004, and the initial thing that we did was to take viruses from throughout this network around the world. All of these black dots indicate places where viruses are collected by this network, of thousands of people worldwide that collect strains of flu. In over 100 countries, these are just the directors of those centers in over 100 countries. And those data are sent to 5 centers around the world for detailed analysis, in Tokyo, Beijing, Australia, the United States, and in the UK. Today, and for the last about 10 years, those data, as soon as they've been analyzed in one of those centers, those data are sent to Cambridge to analyze using this methodology, and within 24 hours, this data is turned around to make antigenic maps that look like this. And we're now going to look at tracking the evolution of these viruses in real time, in this particular case for the purpose of vaccine strain selection. We're going to look at a series of maps like this, which is focusing in on the bottom of this map to show, in fact, what happened since 2002 to the present day. These blue dots here indicate a vaccine strain, and the little dots, a strain that was taken from somebody's throat somewhere in the world in a particular month. This month was July 2002. I'm not going to show you every month in the interest of time, but the next month here, this was enough change that necessitated an update to the vaccine. Fortunately, the previous cluster goes extinct when this particular virus evolves to a new type. 1 year later, it did the same thing, and the vaccine had to be updated again. 1 year later again, kind of hung around there for a couple of years. And then in 2009, it jumped here and has remained here since. The question of why this evolution is happening, why it would jump every year for 3 years and then for the last 4 years not jump anywhere, it's one of these key evolutionary questions that is being studied by a lot in Cambridge and in other places too. One piece of work that led to this, and this was work that was also that was published with all of the WHO network, the people who contribute to this, this was to figure out how these flu viruses spread around the world. It turns out that the type of flu that causes most morbidity and mortality, the H3N2 viruses, they circulate by passing from country to country in Asia around during the year. And every year, the flu we get in Northern Europe, North America, and also in Oceania, it comes from Asia every year. And so actually, if we want to really look at where the selection pressures are from the virus and where to really be focusing surveillance for these H3 viruses, we now know that we need to be looking here. That was the second a little anecdote. The third one that I'm going to give here had to do with some work that was published about a year ago that was analyzing the potential for bird flu viruses, in particular this most nasty type of bird flu, H5, as to whether or not this virus could evolve in nature and start transmitting between humans. Could it cause an influenza pandemic? You might remember the brouhaha in the press around about a year ago as to whether or not this work, which was discovered in a laboratory in Rotterdam and another in the United States, these viruses, if that work should actually be published, or if it should be kept secret because it could be a dangerous bioweapon. A piece of work that went on here in Cambridge was asking the question, really to try to balance the risk of knowing that information, the risk from human nature of using it in a bad way, versus the risk of Mother Nature evolving these viruses and the importance that we should know about this. The work should be published, and research should be done to try to protect. And work here in Cambridge asked questions in great detail, which of the mutations that would be needed to make these viruses airborne transmissible are already out there? Might the remaining mutations actually evolve within human hosts who are infected with H5. It's only been about 1,000, so it would have to be a quick evolution. And are there any actionable items to reduce the risk? Turns out that 2 of those mutations that were needed were already out there. But a number of factors that could slow down the evolution within host were actually outweighed by factors that could improve it. And there are a series of actionable items that can be done to reduce this risk from nature. And indeed, this risk analysis that we did was one of the main things that was responsible for this work being published for the US government and the Dutch government changing its mind over whether this work should be published. There were a couple of other things as well. The last thing I'm going to mention concerns dengue. This is a virus that causes a great deal of issues in tropical countries, a little bit like malaria. This is a consortium of 20 of the best dengue labs around the world that we've pulled together, work led by Leah Katzelnick, to make this an antigenic map of the dengue viruses showing how these relate to each other. I said before that these sorts of antigenic measurements, they're very difficult to interpret, and people have not done that accurately for the dengue viruses. And as a result of doing this, we now know that antigenic variation among the dengue viruses is much more important than people thought. People previously thought there were 4 major antigenic variants and that was it. We now know that that there is variation within these variants, and that the difference between these different variants is only about as great as the difference within the variants. This has great implications for what strains of dengue one would put in the vaccine, what effect the introduction of the dengue vaccine might have on the further evolution of the pathogen. We're doing a similar thing with enterovirus 71, a polio-like disease that also infects millions of people in Asia. There are the little 4 caveats that I wanted to mention, and I think that I will leave it there, except to say this is a pan-university effort with broad funding as well, and also many people from around the university involved, and also a lot of hookup with the different communicable disease and infectious disease initiatives around the university, the Cambridge Infectious Disease Initiative, Center for Global Health Research, Thrive and Cambridge in Africa, with people really working extremely well together. We still are in an evolutionary process, though, of figuring out how all this fits together and how it fits in together with this broad public health initiative. It's an exciting time. Thank you.
Thank you very much, Derek. Time for 1 or 2 questions.